Welcome back, everyone. We had a short break in the action there. My name is Gustavo Ramirez. I'm a microbial ecologist at the California State University in Los Angeles, and I am enjoying the last dive of the first leg of our Octopus Odyssey expedition here. Wow. Let's just take some time to enjoy this close-up of this majestic deep-sea octopus. This is a male. I am learning how to sex this animal, so having spent the last 20 years of my life looking at bacteria under a microscope. So this is a male, and we have a very close view here of its right eye. And yeah, amazing animals. Guardians of the abyss in my mind. Looking into their eyes, I feel like the entire ocean stares back at me. Unbelievable beauty here.
It's almost like you walked into something, scraped on.
Yeah, yeah. And again, we've seen that with skates, right? Are there chromatophores reacting to our light set?
No, not really.
I wonder what would happen if we dim our lights for 5 seconds and then just turn them back on.
I am Carlos was trying to.
Something I want to try, or
Ask the pilots.
yeah,
Do you want to switch the lights off?
yeah. What are the chances we can switch as many lights off as we can for 5 or 10 seconds and then just turn them back on? Trying a bit of a impromptu experiment here. Cool.
Nice and moving.
Tell the people what you do.
You just leave that for like 5 seconds.
Yeah.
Leave that
Yeah.
work out a little better. There you go.
Is he turning darker? No, right?
Or is it
Must be the shadows. Yeah.
He's just using a movable light right now. Try to get it just right. Oh, actually, I can show that. HD2 or 1.
Again, this amazing view is basically the ROV having dimmed its lights just to see what kind of behavior that might elicit on this majestic deep-sea octopus. Guess there's very little light at the seafloor at this moment, and that's intentional. We're going to let this darkness keep on going for a few more seconds here before we turn the lights back on. Just to see how this animal reacts to lights coming on all at once, rather than a slow spotlight slowly imbuing the field of view there. So I'm not sure if I'm seeing any changes.
Yeah, so the
I
light
guess
we have on is the light on the manip pointed at it. That's the view from the manip.
It's playing.
So briefly here, we will turn our ROV lights back on just to see if there was any differences in the chromatophores.
chromatophore organs?
In the chromatophore organs that octopuses have. Again, these animals live in perpetual darkness down there, and I'm not sure if they ever come into contact with the light world. Probably not. Their ancient ancestors likely found their way into darkness right from the sunlit world. Okay, and he's gone. Beautiful. Wow. That's a lot of drama. Right?
We think the common ancestor of this family actually got into the deep sea from the high latitudes in the north.
Okay.
So if you're living high up, there are times where you hardly see daylight,
Right.
even in the shallow water.
Right.
So maybe going to 365 days a year of dark wasn't
Wasn't
a
that big
deal.
of a deal? Correct. Yeah.
It's cold at the surface, it's cold at the bottom,
Yeah.
and unexploited.
So that was again a fun little impromptu experiment with the ROV lights there on what is the last dive of the first leg of our Octopus Odyssey Expedition there. Again, these animals, they're lined with chromatophores that are specialized cells that can react to light. And we were just curious to see what would happen if we dimmed our lights for a couple of seconds and then turned them back on, see if there was any visible changes on the elicited by the chromatophores of these animals. Hard to tell if anything actually happened, though, right?
Right.
Yeah.
I've got a couple pictures of granulatinate that was photographed from Alvin. And in the one, it looks purple. It's really close to the viewport. And then they turned on the light over the outside the viewport, and it looked orange. I don't think the octopus had anything to do with it. I think it's the angle of the light. There still might be iridophores within the skin that reflect light in different ways. I saw maybe just a hint of that. But I think these guys patch out orange, go through life to full maturity as a beautiful maroon, and then as they get old and decrepit, they kind of turn a pale purple.
Old and decrepit pale purple.
Yeah.
Expert
You know,
commentary from.
The blue hair. They go out to bingo every night. No.
Expert commentary
For dinner.
from the Octopus World Authority, Janet Voigt. Oh, how am I doing with time?
It's like your shift change.
Yeah, we're coming up with a shift. How you doing? Good. Okay, it's all exploration. Just having fun, really. It's incredible. I saw that, man. Yeah, that's very nice. Yeah. Alex made a very simple story incredible.
It's super nice.
Oh my god. I was
That's
telling
cool
him,
rock
I
star,
hope
you
he's
know?
got another victim. I don't want to be the only one. Yeah. He better be doing somebody else's right now. Yeah. So anyway, we've just been flying around Waypoint 1, staying along the contour line, looking at octopus. Janet's here, you know.
We got a little bit of a handover happening here. While we're waiting on that, it looks like our internet's stabilized a little bit, so hopefully our audience at home is having a good time exploring along with us. For anyone who might have just joined us, we are streaming live footage from the remotely operated vehicle, or ROV SuBastian, from about 3 kilometers down, below the surface of the Pacific Ocean, off of Costa Rica. And we are in an area called Dorado Outcrop, which has many of these octopus. And this one in particular is a male. We were able to identify that off of the third right arm being modified at its tip. But there are many females in this area as well that have chosen Dorado Outcrop as a brooding ground. Actually laying their eggs amongst the rocks, amongst hydrothermal flow, warmer water coming up from below the ground. And scientists are interested in this behavior, in the selection of this area, in the geology of this area, the microbe biomes of this area. A lot of work going on here in this expedition. And this is the last dive of the Octopus Odyssey Expedition right now, although we will be returning to this location in December to check in again. We left some experiments, we've left some probes, so still more work to be done here, and lots of work that's already been done that you can catch up on. If you head over to schmidtocean.org, there is an expedition page for Octopus Odyssey, and there are blog posts there written by the scientists on board, which are just fascinating. You can also check out the hashtag OctoOdyssey, Lots of people have been sharing screen grabs and trying to ID some of the stuff that we're a little befuddled by sometimes, and some cool posts from the scientists as well, looking at some of the specimens that made their way up to the ship. So lots out there to learn and to catch up on if you're just joining us. And I think our science handover is done, so I'm going to hand it on back.
Hello everyone, this is Maria from University of Costa Rica. So I will be with you this hour. So here we continue looking at the Dorado seamount with this beautiful octopus here, swimming. Hola, muy buenas tardes a todos y a todas. Les habla María Isabel Sandoval de la Universidad de Costa Rica, de la Escuela Centroamericana de Geología. Estamos en nuestro último buceo el día de hoy de esta expedición tan maravillosa que hemos tenido en las zonas profundas de Costa Rica, en el Pacífico Norte. Y aquí estamos observando un hermoso pulpo nadando. Este es un macho. Hemos tenido unas excelentes tomas de estos octopus, de estos pulpos. Bueno, y cerramos con broche de oro esta expedición, viendo estas imágenes en uno de los sitios más impresionantes de los montes submarinos acá de Costa Rica, en el Pacífico. Este sitio ha sido en particular, nos ha dejado muchísimos descubrimientos. Hemos visto muchísimos pulpos en este sector asociados a las aguas termales con salidas de agua caliente. That's great. Yeah. Me quedé hasta sin habla de esta forma de nadar que tienen este pulpo acá, este macho. Estos organismos son del género Octopus. Y aquí tengo a la par a la experta aquí en la sala de control, admirando esas hermosas imágenes. Bueno, hoy termina la expedición, pero tenemos en línea en la página del Schmidt Ocean muchísima información sobre esta expedición. Les recuerdo que volveremos para diciembre con más de esta expedición. Vamos a volver a algunos sitios que dejamos algunas pruebas y algunos experimentos, a recoger estos experimentos y también a continuar con algunos objetivos que tal vez nos quedaron, o algunas muestras que tal vez quedaron pendientes. Y así que esperamos que nos vuelvan a seguir, ¿verdad? Y a acompañar durante esta hermosa expedición. En los sitios visitados, les recuerdo que tuvimos varios, 2 sitios no antes explorados, que fue al este de Caballito. Tuvimos el sector de Caballito también, que estuvimos el día de anteayer, estuvimos en un seamount llamado Tengo Sed el día de ayer. El Dorado estuvimos, ya esta sería la tercera vez que vinimos. Y ¿cuál otro me falta? Creo que estamos con, sí, creo que esos fueron los sitios visitados. Y un seamount que tampoco habíamos ido, que es el seamount de Fuente. Todos estos videos los pueden ver en YouTube. Por si se perdieron alguno de los buceos, entonces pueden volver a revisarlos y a verlos. Además de los highlights que tuvimos acá o de los principales descubrimientos, es que sí tenemos asociados principalmente a esos musóctopos a los montes submarinos que tienen salida de agua termal. Vimos algunas otras especies como graneledone en sitios de más bien de recarga de agua fría, y en los sitios de salida de agua caliente o agua más caliente más bien tenemos los pulpos asociados a este género específicamente. Al lado mío hoy también tengo como observador al estudiante de doctorado Rob Perrin de la Universidad de Calgary. Rob ha trabajado muchísimo en esta expedición y sobre todo con los datos de flujo de calor y medidas de temperatura en todos los sitios. Ellos son, él junto con la doctora Rachel Lauer, trabajan con la parte geofísica y lo que está pasando a nivel hidrogeológico y la interconexión que tienen estos seamounts y el sistema en sí.
Stayed along that ridge, and it seems to be nice and rocky and stuff, just looking out for whatever.
Bueno, aquí estamos haciendo algunas tomas de este pulpo macho, pero en sí seguiremos recorriendo un poco del sector para que conozcamos más de este sitio tan espectacular que es el Dorado. Bueno, para comentarles también en qué estuvimos trabajando los investigadores. Teníamos diferentes grupos de investigación acá a cargo. Bueno, el grupo de biólogos que trabajan específicamente, los especialistas en pulpos, que serían la doctora Janet Voigt, que es experta mundial de pulpos, o la experta mundial, por decirlo de alguna manera, y también su colaboradora en la parte genética, que es Holly Lutz. Y posteriormente tuvimos, como les mencioné, al equipo de geofísica. También teníamos el equipo que trabajaba sobre todo con la parte del microbioma, de microbiología, que estuvimos escuchando a Gustavo. Por cierto, tenemos un video de un poco de la motivación de Gustavo en estos estudios y demás en el canal de Schmidt Ocean. Y después está, bueno, en el caso de mío, trabajo con sedimentos principalmente y con la parte de microfósiles. Además tenemos a los investigadores de la Universidad de Costa Rica trabajando específicamente corales negros, corales octocorales, muestras de agua para acidificación oceánica. También tomamos muestras de rocas para el análisis geológico. Algunos colegas, bueno, el colega Sergio con la parte de oceanografía principalmente, y muestras también muestras de agua. Y el equipo de microbiología que estaba constituido principalmente por Gustavo y Miguel Semedo. Es muy importante estudiar todos estos sectores, este sector tan desconocido, esta Costa Rica desconocida, como hemos dicho, lo hemos dicho varios acá en esta transmisión, para conocer lo que tenemos como país y también para poder protegerlo. Sí, este sitio o sobre todo esta expedición ha sido muy valiosa desde el punto de vista no solo científico, sino la parte de divulgación con la que trabaja el Schmidt. Es impresionante porque podemos ver imágenes del fondo oceánico en vivo, podemos interactuar con ustedes como público interesado, y para nosotros esto es sumamente valioso porque conocemos no solo Costa Rica, sino todo lo que es fondo oceánico, que siguen estas expediciones. Posteriormente tenemos expediciones en Galápagos, por ejemplo, y, y esto nos permite conocer un poco más de estos lugares que no podemos llegar fácilmente, ¿verdad? Solamente con equipo muy especializado y muchos miles de dólares de presupuesto, ¿verdad? Que tal vez nuestros países no permitiríamos tener este tipo de equipo en el país, ya que es sumamente caro. Quedan algunos remanentes de pilos lavas acá, vemos las fracturas las fracturas de enfriamiento en algunas partes, pero aquí tenemos principalmente lo que son bloques o caída de bloques y un poco de sedimento. Este monte submarino ha sido datado en 23 millones de años, estas lavas. Y también tenemos asociados sedimentos, rocas sedimentarias también del Mioceno. Can we take a look on that one?
Upper one?
Yes.
You guys want to land and look?
Yeah, please.
So we get them in the line correctly.
Are you interested to take a T probe here? A T probe here in this fracture maybe, close to the octopus.
Yeah, we're all out of temperature markers. Or do you want to take the temperature?
Yeah.
Yeah,
Yeah, okay. Yeah. we can take the temperature in one of the fracture. Bueno, este sitio ha sido visitado desde el 2013 y todo lo que es el estudio de flujo de calor se describió desde ese momento y se había observado esta cantidad de pulpos acá, que había sido una observación bastante interesante, y desde ahí nació la posibilidad y la oportunidad de, de, de poder promover un proyecto nuevo y volver a este sector, y sobre todo con la pregunta de investigación relacionada con este hábitat tan interesante para los pulpos. Desde ahí, bueno, pues ahora nos dedicamos también a seguir tomando más datos de temperatura, de flujos de calor. Aquí tomaremos posiblemente un dato de temperatura si logramos hacerlo. Aprovechamos a veces estas salidas o donde están viviendo los pulpos. Aquí vemos una fractura donde está uno un poco escondido detrás de esa esponja. Aquí se puede observar un tentáculo. Thank you. En este sitio estamos a una profundidad de 3030 metros de profundidad. En muchas de las ocasiones vemos a los pulpos incubando sus huevos, y en algunos momentos también a veces se observan lo que son se observan algunos machos también. Por ejemplo, el que vimos antes nadando era un macho.
You could keep the forecast on her, I'll just use the sit.
Bueno, les sigo comentando los estudios que tenemos, el equipo de investigación. La doctora Beth Orcott, que es la jefa de la expedición, ella también trabaja con lo que es la parte microbiológica, pero en asociación con las rocas y con el agua, además de Tim, que es estudiante de doctorado, bueno, postdoctorado, perdón, realiza un postdoctorado. Y después tenemos dos artistas que están, bueno, que han estado inspirándose en estas bellas imágenes y en el trabajo que estamos realizando para también pues, realizar sus pinturas. Tenemos a lo que es al artista argentino-costarricense Carlos Hiller y al artista estadounidense Michael Drudge. Un saludo para mis colegas de la Universidad de Costa Rica, la Escuela de Geología, que me están escuchando. Aquí vamos a hacer una toma de temperatura. Bueno, aquí estamos midiendo una temperatura 220 como máximo. 234, veo ahí en pantalla. Y se pegó. He wants to come. Hey. Oh, oh. Bueno, a veces sucede eso, pegan los tentáculos a alguna de las pruebas que se realizan o al equipo. Yes. It's okay. Thank you. Yeah. Bueno, aquí la máxima temperatura fue 234.
Alright, what's coming in? Continue.
Yes. Yeah, we'll continue.
Alright. Come out the wall. And come again. That's cool.
Y los organismos bentónicos principales tenemos octocorales, esponjas, como estos que se observan acá. A veces podemos observar unos briozoos. Estas esponjas de vidrio han sido bastante vistas en el sector del Dorado. Se ven como hermosos jardines. Un saludo a mi hijo, Emil, que me está mandando corazones. Muchos corazones también, amor. Me pareció ahí, lo que teníamos era un pepino de mar. Cuando se ven estos parches oscuros sobre la roca, esos ya son esponjas muertas, ya recubiertas de sedimento, también brosoarios. Si miramos dentro de esas esponjas de vidrio, encontraremos algún tipo de camarones que viven ahí dentro y quedan atrapados por toda la vida con su pareja. El artista Carlos Hiller se inspiró en una de estas tomas para hacer una pintura. La pueden observar en la página de Facebook. Es muy bonita y dice mucho de esta interacción. Bueno, acá tenemos otro pulpo. Tenemos dos, parece. There are two, right? Two? Yeah. Estas estrellas que tenemos acá son picnogónidos. Son estrellas que tienen de 6 a 20 brazos. Y estos pequeños octocorales negros de la familia de los ostoloníferos se han observado bastante asociados a grietas. Hemos notado algunos de distintos colores, blancos, a veces con tonos más rosados. La doctora Odalisca Bride, que se dedica a esto, ha trabajado con este grupo y seguirá investigando. Como les contaba, la doctora Bride ha determinado para el Pacífico Oriental tropical 43 especies y para Costa Rica específicamente 23. Así que hay todavía muchísimo por descubrir en estos sitios aún inexplorados y con muestras únicas desde diferentes puntos de vista científico. Se encuentran muchísimas cosas todavía.
Sit down so you can see wider view, so you can see how far away you are.
Bueno, aquí se observan más estoloníferos. Aquí tenemos una buena toma. Parte de nuestro equipo científico y jefe también de esta expedición es el doctor Jorge Cortés, que dedicó su vida al mundo marino y la exploración de los océanos y dar mucho conocimiento al país desde el punto de vista principalmente somero y a partir de cierto momento pues Jorge inició lo que es esta gran aventura hacia el mundo profundo, o eso el océano profundo e inspirar a muchos de nosotros como costarricenses a trabajar con a estas profundidades. Así que ha sido o será o para nosotros es de una persona que nos ha motivado y que nos enseña cada día y sobre todo a apreciar el fondo oceánico, pero también a protegerlo de alguna manera. Y también otra cosa muy importante es trabajar desde el punto de divulgación para conocer estos sitios, así que tenemos en vista algunos proyectos desde la Universidad de Costa Rica y la Universidad Nacional para dar a conocer un poco más sobre esta expedición y sobre expediciones pasadas, todo lo que es esta Costa Rica desconocida. En este momento nos encontramos a 3025 metros de profundidad en el monte submarino El Dorado. Y estamos hoy en el sector noroeste de este monte. Les recuerdo que, bueno, hemos venido, ya esta es la tercera vez que estamos por acá durante estos días en esta expedición y hemos tomado temperaturas máximas, salida de agua hasta unos 12 grados. Esta es una esponja bastante grande y tiene unos crinoideos ahí pegados, unos 3 crinoideos que vimos. Estos crinoideos, estos organismos son del filón Echinodermata y son organismos muy antiguos que tienen sus orígenes de unos 200, 300 millones de años hasta la actualidad. Así que nos siguen acompañando. Lo mismo que las esponjas, son organismos con un linaje antiguo muy importante, inclusive mayor que este tipo de equinodermos. There. I think what's an octopus here. Here, an octopus, no?
That's where we were a little bit ago. Oh, yeah. Down a little bit?
No, yeah, no. I thought it was here. It's a sweet cucumber.
Yeah.
Here. This one.
Oh, yeah, 'cause we made
Yeah.
a big circle around.
Ah, okay. Bueno, un saludo de feliz cumpleaños a mi tía Irma, que hoy está de cumpleaños. Con estas bellas imágenes, un regalito desde el fondo oceánico, tía. Este sector es bastante empinado en el Dorado. De las partes se observa muchísimo como este tipo de caída de bloques y algunos remanentes de los pilos. Aquí tenemos una medusa.
Can you share with the audience that the cold can that we recovered is from 1971?
Really? Yeah, sure.
100% sure now if this is accurate.
Wow. Bueno, Gustavo me está contando que la Coca-Cola que se recolectó en este sector, como a las 12 mediodía, es de 1971. En parte triste encontrar lo que es ese tipo de basura acá. Imagínense desde hace cuántos años tiene esto acá. Y bueno, fue recolectada para interés científico para el colega Gustavo. El análisis de la microbiología en esa lata de coca, pero pues en realidad es tristísimo pensar que llega basura hasta acá, a 3000 metros de profundidad. Okay, just to let you know, the age of the Coke is from 1971. Las esponjas, bastantes esponjas asociadas a crinoideos en este sector, estos jardines, estos de mis tomas preferidas, este montón de exactinélidas con estos crinoideos. Me siento así como si estuviéramos en el Cámbrico, en el Ordovícico. Briosos. Pareciera una toma desde hace unos 300 millones de años, estas tomas así. Las dimensiones de esos octocorales son grandísimos. Esos octocorales son organismos que pueden tener hasta miles de años de crecimiento. Hasta el momento la basura que hemos encontrado ha sido principalmente metal en este sector. Plástico, pues no, pero tenemos uno de los objetivos de investigación es estudiar las partículas de microplástico también, que lo realizan investigadores del CIMAR. Entonces, para posteriores estudios sería importante saber cuál sería ese porcentaje de plásticos que hay en esto, o que llega hasta el fondo oceánico, hasta unos 3.000 metros de profundidad, y conocer realmente este dato, ¿verdad? Les recuerdo que aquí las tasas de sedimentación son muy bajas en el fondo oceánico. Podemos tener desde 1 centímetro cada 1.000 años, inclusive más viejo. Así que el análisis se tiene que hacer realmente en los primeros centímetros para poder encontrar restos de plástico. Aquí podemos observar algunos pilos más formados. Vamos a ver qué es esto. Ni idea. A ver si los biólogos nos ayudan. A veces tenemos aquí en el cuarto de control algunos biólogos que nos ayudan a ir describiendo un poco, pero aquí estamos solo Rob, que es geofísico, yo geóloga, y Gus, que es microbiólogo, así que no somos los especialistas para esto. Algunos organismos todavía nos parecen increíbles, no los reconocemos. El tamaño de ese octocoral impresionante. No tenemos los láser puestos, pero con solo verlo a la escala comparado al crinoideo que estaban ahí, o los ofiuroideos, puede medir más de 2 metros. Vean que a pesar de que el Dorado es una de las estructuras, es una estructura volcánica relativamente pequeña, comparado a algunos otros sitios que hemos estado visitando, como el Tengoset, que es uno de los más grandes, aún así se recorre pues lentamente, ¿verdad? Para ir observando, viendo si hay algunas asociaciones interesantes aquí, tanto faunales como con la interacción de flujos. Se observa desde el punto de vista geológico también, y, y por eso los buceos toman generalmente 1 hora cada 100 metros para un recorrido de 100 metros o así. A veces cuando tomamos muestra pues se dura muchísimo, ¿verdad? Dependiendo la muestra que se vaya a extraer. Y, y hoy nuestro último buceo va a ser de 16 horas. Sí, hoy se tendría un buceo hasta las 6 de la tarde, sí, no me equivoco. I think for today we have 2 hours left for this dive. It's a dive of 16 hours. Y les agradecemos a todos por haber estado, todos y todas por haber estado conectados, aprendiendo, conociendo el fondo oceánico, haciendo comentarios, preguntas. Una de las maneras que tenemos de reconocer cuando hay salida de agua o flujos más calientes o una descarga de aguas en estos montes ha sido justamente ver las rocas limpias, ¿verdad? Sin sedimento. Y acá estamos viendo muchísimo sedimento. Entonces, así que, como vieron, hemos estado midiendo temperaturas o medimos algunas temperaturas bajas relativamente, 2 grados.
Figure out says you haven't heard anything different as far as recovery from normal times. So.
Go until 7. Where?
Here, next to that sponge.
Oh, the drippy guy?
Yeah, can you zoom in on that?
Oh, yes, we can, Howard.
Right. I'm just thinking, like, there's all this eastern wasting disease that goes on, and it's not this, but this is the first weird one we've seen.
Yeah.
Are we allowed to collect?
Of course we are.
I'm not in charge, though. But, and they're, wow, that is actually, hang on. What are your thoughts? It's pretty big.
Should we have the lasers?
Yeah. Okay. Yeah.
Yeah, those are gonads that are coming out down its arm on the front arm. And when they start to die, that can happen. Jeez, that's not good. Would that fit in a river?
I think so, yeah.
It's so big. We can't. Yeah.
Yeah.
Yeah, it's not,
Too big.
that would be great.
Yeah. In a coral quiver?
Yeah.
Yeah.
Yeah, and it can kind of get smushed in there if it needs to.
Yeah.
Um,
So see this bulge right here?
Yeah, we got
Yeah, I see running that. And then what is it again?
La doctora Holly Lutz está interesada en tomar esta muestra de estrella de mar, por lo tanto vamos a estar acá unos minutos tomando esta muestra. vamos a colectar esa estrellita.
¿Es estrellita?
Ujú.
Ok.
Sí. Llegó Holly aquí a abrazar.
Holly, le digo.
Sí.
Ok.
Yo no había colectado nada, pero no dice que ella está interesada para estudiar las gonadas de esta.
Ok.
Entonces, y ahí vamos a estar un ratito.
Ok.
Entonces, si quiere ya le dejo el
Dale.
sign.
Igual si quieres seguir grabando.
Familiar with SOI disease? Mass mortality. They thought it was a virus for a while, but it's not. It's what
So thank you so much, everyone, for being on all these days with us. And so my colleague Sergio Cambronero will be with you this hour left. He's from the National University here in Costa Rica, from the Physics Department. Muchísimas gracias a todos y a todas por estar conectados durante todos estos buceos que hemos realizado. Y los dejo con mi colega Sergio Cambronero de la Universidad Nacional y del Departamento de Física. Muchísimas gracias.
Pues ahorita nada más explorar y muestrear. Ya,
Se finí, exacto.
¿Y cómo vamos con a dónde está la No, la muestras.
Those are the ones we have marked.
I think we should say goodbye.
All right, we got new folks settling in for a new watch here. Still got Tyler on controls, it looks like. And then J-Rod's going to be in co-pilot seat right now on the manip. And I just got back in here, so thanks for Josh sitting in for a little while, keeping the stream going.
Yeah, can you turn the lasers on one more time? I'm sorry. Okay. Yeah, we're good then. You can go. Thanks.
Thank you to my colleague Maria Sandoval from University of Costa Rica. My name is Sergio Cambronero, and I am a marine biologist and have been working in physical oceanography for the last 8 years at Universidad Nacional in Costa Rica. And I will be joining you for the next hour. Hola a todos y a todas. Mi nombre es Sergio Cambronero y las voy a estar acompañando por la siguiente hora en esta transmisión. Soy funcionario de la Universidad Nacional del Departamento de Física, biólogo marino de formación y he estado trabajando en oceanografía física durante los últimos 8 años. Así que para mí será un placer estarlos acompañando y si tienen alguna pregunta, algún comentario, pues la dejan por acá en los canales de Facebook o YouTube y con muchísimo gusto los vamos a estar leyendo. So greetings everyone. We've had a pretty amazing last dive. Unfortunately, last dive, but just for this expedition. Good thing is that we will have another one if everything goes accordingly to the plan. We'll have another one in December. So this has been a really impressive dive because we have been able to see, oh, that shrimp just was like, huh, step away. But yeah, we have been getting great observations of octopus. We were also able to get some samples, and there is a particular sample that we got earlier in the morning. That was a wood piece or a log with what we think are some sea lilies of genera Xyloplax, and these are particular echinoderms that we are very interested. And when I say very interested, I say really interested. So we have been going back and forth on how to preserve them the best way possible, because we might have an amazing finding here while we sample this sea star. So again, if you have any questions or comments, please leave them in the chat, and we'll be reading them here. So no chimeras today, but yeah, hopefully we'll find another one. I think that's too much to ask, but yeah, fingers crossed.
Should be a pretty rigid animal. It's not gonna fall
Yeah,
apart.
just someone to type it slow. That's more fun.
So we have a comment or a question whether the wood was petrified. So by looking at the pictures, I was actually on a ship to shore event, so I was not actually witnessing the amazing finding, unfortunately. So I haven't been able to actually see that good, but it didn't look petrified. It looked kind of fresh, relatively, of course. But yeah, it still looked, with a wooden collar. So yes, I wouldn't think that it was petrified yet.
But we collected that, correct?
The wooden one?
Yeah. That's very cool.
It's really cool because those sea lilies might be a whole thing.
Yeah.
And we were trying to see how to preserve them.
This could go in a bio box if needed.
I mean, that's the thing, right? Is
Of course, that would be way.
Yeah, I think
like whale falls get a lot of attention. People are familiar kind of with the concept of whale falls. So if you're not, right, like if a whale dies and it falls down, all of a sudden you have this oasis of food for all sorts of organisms that will devour that whale over a long period of time.
What level?
3 would be fine if that's okay.
Okay, let me just set this down then.
But wood falls are their own thing, right? You have whale falls, which I think they're probably a little more charismatic, right? It's this this environment built upon a dead whale. But wood falls are an important thing in the deep sea as well.
Totally. Yeah, I agree. Definitely, Alex. And we've checked. Small world. I met several persons at a echinoderm conference like 7 years ago in Costa Rica. And one of the main conference that really left an impact on me was one of these sea lilies being taxonomically controversial because some people consider them as being one specific group within the echinoderms, and some other people will classify them as part of the Asteroidea group. So again, as I was saying, small world. Turns out that guy giving the lecture, his name is Don Janis. He co-authored that paper with Dr. Janet Boyd here. So yeah, turns out that we have Dr. Janet is not also a specialist in octopus and cephalopods, but also has experience with echinoderms, and especially with this particular group that are called sea lilies. Very enigmatic because they're echinoderms, but do not share some of the main characteristics.
Very cool. Excited to get that up here.
Yeah, totally. Excited to see. Y para, bueno, comentaros un poco lo que estábamos hablando en inglés. Parte de lo que vimos anteriormente en este buceo y que estaba comentando Alex fue que usualmente a estas profundidades tenemos a veces observaciones de esqueletos de ballenas, por ejemplo, o de, de cetáceos, pero también no solamente estos esqueletos pues se hunden desde arriba hacia abajo y son como representan un sustrato para que crezcan animales, sino que también pueden ser pedazos de madera que cumplen más o menos una función similar, ¿verdad? Que es como sustrato de la superficie que se hunde eventualmente en el fondo marino y pues que es colonizado por varios organismos. Entonces el día de hoy en la mañana pudimos observar un pedazo de madera que tenía muchísimos organismos asociados y dentro de ellos un grupo en particular que es un grupo muy enigmático que se le llama margaritas de mar y básicamente son emparentados con los equinodermos, o sea, las estrellas de mar, los ofiuroideos, los pepinos de mar, pero que sin embargo ha habido un debate y por decirlo así una controversia cómo es que se tienen que clasificar, si son asociados a las estrellas de mar o si son un grupo por sí solos. Entonces, la ciencia es algo que está en constante evolución, siempre, pues nunca existe una palabra final, siempre todos son hipótesis que están en constante revisión y el día de hoy creo que pues podemos aportar más y vamos a aportar más, no tengo duda, una pieza más para resolver ese rompecabezas. Así que bueno, pues muy emocionados por esos organismos que recolectamos hoy. Y si no los vieron, pues les invito a que vean los buceos, las grabaciones del Schmidt Ocean Institute en el canal de YouTube y en Facebook. Este es nuestro último buceo de esta expedición. Para los que nos sintonizan en vivo, pero lo que les puedo comentar es que a pesar de que pues, a todos nos pone tristes, ¿verdad? De que todo se acabe y sea el último buceo, vamos a regresar, si todo sale de acuerdo a nuestros planes, en diciembre a esta misma área. Así que para que se mantengan atentos a las diferentes redes sociales de las instituciones involucradas, como la Universidad de Costa Rica, la Universidad Nacional, el Laboratorio para las Ciencias de Bigelow y el Schmidt Ocean Institute, por supuesto, porque ahí pues vamos a seguir dando actualizaciones. Pero bueno, sí, no, aparentemente no. Si todo de nuevo sale al plan, estaremos regresando acá en diciembre y lo que les quería decir justamente es que aparentemente hasta el momento pues todo va sobre ruedas, como decimos, viento en popa para esa segunda expedición. So we have a question, well, several questions in our chats here about the Sea Star, and as my colleague here, Holly Lutz from Scripps Research answered on the chat, looked like that Sea Star was in its last moments. So yeah, we are thinking, and that was what I would call a sample of opportunity, because it was pretty much just not very active. So you could tell that you can tell that by the inactiveness or the lack of movement of their little tube feet that you see below the arms, and also because of the position that it was just hanging. So again, special thanks to Dr. Holly Lutz from Scripps Research that is here in the control room, diving, leading this dive and taking care of the video annotations, which are, I would say, the most important part of all these operations. And we also see some questions whether we've seen any shimmering water during this dive. So the answer is yes, but only in few spots at the very beginning. And I think we got the highest reading, about 10 degrees, right? I think. So yeah, 10 degrees. And we also saw some octopus and brooding octopus around this area. So yeah, it has been an incredible dive. And this, where we at right now, is the northern part of Dorado Outcrop. And in our initial dives, we have been exploring this area, but more towards the southern end of this feature. And today, for the very first time during this expedition, we came here to find very incredible surprises. So thanks again for all those questions, and yes, keep them coming. We'll love to also know where you're watching this from. So yeah, probably nighttime for some of you. And yes, and again, if you have any questions, comments, put it in the chat. Yeah, I don't think it's a gravity.
Anyone who's just tuning in, this is live footage, believe it or not. You were looking at an outcrop, Dorado Outcrop, a little rocky outcrop here about 3 kilometers below the surface of the ocean, off the west coast of Costa Rica. We're just watching this fish. This footage is coming to you by quite the way. So this is a remotely operated vehicle named SuBastian. It's being piloted by pilots up on a ship on the surface, the research vessel Falcor 2. So they are sending commands down to the ROV, remotely operated vehicle, over, well, let's see, we're 3,000 meters down, 3,030 meters. We've currently got about 3,083 or so meters paid out, though. So it's going. We're sending the controls, the commands down this long tether to the ROV. ROV's doing what the pilots are directing it to do, but the footage is coming back up that umbilical to the ship, where we can see it here in mission control, and we can also send it out to you. So the ship is fitted with satellite internet, and it's playing pretty well for us today. Sometimes it gets a little fuzzy here and there, but yeah, it's doing great right now, and we are able to bring you live HD footage from the deep sea as deep sea science is being conducted. Now, the reason we're here is that this outcrop has been found to be the home of many an octopus. And these octopus aren't just hanging out. They're actually using a feature of the outcrop. They are brooding here, which means sort of tending to their eggs. We may see some female octopus. They're sort of flipped upside down. They look like a strange umbrella or something. And those octopus tend to have eggs underneath them that they are brooding, that they're taking care of. And scientists are interested in the areas that these octopus are using for this behavior, which seems to be related to hydrothermal venting. where warm water is actually coming up from underneath the surface of the ground here. And so scientists have been investigating that venting, the octopus, the other animals in the area, the microbes in the area, the sediments in the area, just trying to get an overall picture of everything involved in this very unique find, and trying to determine how unique it might be. Is this something that we can find elsewhere? And during this expedition, we have actually found another little area on another feature nearby where octopus were exhibiting a similar behavior in similar hydrothermal flows. So what's also exciting is, even though this is the last dive of this expedition, they're actually going to be coming back here. We're going to be back in December for part 2 of this Octopus Odyssey. And we're going to pick up some experiments we've left behind. We're going to see if anything's changed, more exploring. And of course, we welcome all of you to explore along with us.
Thank you, Alex. And we are currently seeing what looks like a fishing line. So a clear sign of pollution coming up from human activity. A good reminder, well, maybe not good, but definitely a clear reminder that our impact extends even to these remote areas. And I will also like to send special greetings to all the people reporting from Australia, Germany, Costa Rica, West Palm Beach, Portugal, California, and even New York. So special thanks to all of you for enjoying this live stream. And as Alex was saying, we're exploring around this northern region of the Dorado Outcrop. Y para los que nos escuchan en español, un gran saludo. Por ahí veo a varios colegas, incluida Sileni, pura vida. Espero que estén disfrutando este buceo, nuestro último buceo, lastimosamente. Y para mí es un enorme placer y un honor estar acá en representación de todos y todas las personas que amamos el océano en Costa Rica. Y ojalá que también en un futuro, y estamos, yo pues particularmente y muchas de las personas que estamos acá, muy comprometidos a seguir abriendo estas oportunidades para que más personas se puedan sumar a este tipo de expediciones. Y bueno, acabamos de ver una línea de pesca que es una clara señal de contaminación debido a actividad humana, lo cual nos recuerda pues de que nuestro impacto se extiende incluso hasta estas áreas tan remotas. Así que pues no es una imagen bonita. Anteriormente también vimos una lata de Coca-Cola, lo cual es también bastante triste de ver, pero bueno, parte de todos estos buceos es justamente poder tener esta evidencia y que usemos estas imágenes para demostrarle al mundo también no solamente lo bonito y lo maravilloso de estos ambientes profundos, sino que también demostrar de que tenemos que cambiar de una u otra forma la gestión de nuestros residuos en tierra para que no lleguen a contaminar a estos lugares tan prístinos. Así que bueno, un saludo especial, y de nuevo, si se conectan desde Costa Rica a otras partes de habla hispana, por favor déjenos saber en los comentarios desde dónde se conectan y por acá los estaremos leyendo. Y pues claramente si tienen alguna pregunta o comentario, también con muchísimo gusto por acá estaremos respondiéndoles de la mejor manera posible. So indeed, there was a fishing line, the thing we just saw a few minutes ago. And that's, I would say, clear evidence of the activities we have up here in the water column. Actually, during this last 20 days that we have been navigating around this area, we have encountered and coming across many fishing vessels. So this is definitely a very productive area, not only at the bottom level, but also in upper water column, and specifically this area is under the influence at a specific time of the year, under the influence of the Costa Rica thermal dome, which is an oceanographic feature, pretty much provoked and because of the main geostrophic currents, particularly the north equatorial countercurrent. When it hits the continent, it deflects northward, and it creates a gyre that is persistent, and that it moves and migrates seasonally across the year. So this is an offshore upwelling system, and it is actually really important not only for fisheries, but definitely for other important species. For example, the blue whale has there is evidence that this is one of the most important feeding grounds for blue whales that come and migrate from the north down to these latitudes, and they feed because there's a lot of krill and a lot of food for them here. So again, this area of the Costa Rica thermal dome is also really productive in terms of fisheries, and probably this is the reason why we have been seeing also part of this anthropogenic pollution deep down here at 3,000 meters deep.
Kind of a fun topic popped up in chat here about latency, right? Because I mentioned that the pilots are sending commands down to the ROV, and the ROV is beaming footage and data back up. And ultimately it's almost 6 kilometers of umbilical it's going through, but it's fiber, so that's all traveling basically the speed of light. That's I'm not going to say it, infinitely small. Couldn't do it today. But J-Rod was actually just telling me that years back there was ever so slightly perceptible lag between when they would do things here and SuBastian. This was years ago. And that wasn't in the fiber of the umbilical, but it was once it got onto the ship and it makes its way through the network. And one of the imperative things is that there not be a lag. And so it's actually down to the point now where it is not perceptible to our eyes. So yeah, it's just great advancements in technology for that. Where we can send a signal down 6 kilometers, get stuff back up 6 kilometers, and not have a perceivable lag or latency for our pilots here.
Totally. Not to mention also the minimum lag between what we see here and what people are seeing on their screen.
Yeah. At home, it's a different thing, but for us here.
Yeah. So what's the lag between here and what they can see in their
Really
screens?
depends. Yeah, it could be
Half a minute. So yeah, it's still pretty good.
Yeah. The audience at home is definitely getting more lag, for sure. But on the ship, it's imperceivable.
That's cool. So part of what we've been talking during these last days of our dives and of this expedition among the science party and also with the SOI team is what have been our favorite highlights. So for example, for me, particularly has been the chimera we saw yesterday, among many other observations. But yeah, we're talking here, and I was talking with my colleague Holly Lutz, that it will be a great thing for us also to know what the audience thinks, and you guys watching us at home. What have been your favorite highlights, your favorite observations of this cruise? We know that some of you have been watching these dives following the expedition. So let us know in the comments what have been your favorite moments, your favorite creatures while we watch this beautiful jellyfish? Yeah, it looks like a spaceship. Amazing colors.
You know, it occurs to me that they can figure out their own latencies because we timestamp the video.
Oh,
Yeah.
nice.
So if you've got access to you know, like a internet synced clock or something like that, that's nice. You can compare that to the timestamp and see just how long it's taking for the signal to get from Falcor 2 to you, wherever you might be watching.
This is my favorite moment. Yeah, way to go. So what has been your favorite moment, Alex?
Oh, the Chimera was pretty cool.
Yeah.
Chimera was really cool. Honestly, I've enjoyed a lot of the cephalopods, right? Like we've had the octopus, but we've also had squid. And even just seeing like squid at the surface during recovery and stuff has been
Yeah.
really cool.
Totally.
They come up and you'll see like a little shoal of them, or I'm always pushing for a squad. You'll see a whole squad of squid in the lights of the A-frame when we have the ROV out, or if we're doing even the CTD casts, the light from the J-frame there on the CTD, the squid come and kind of hang out there. And I've always been fascinated with cephalopods, so
Okay.
cephalopods on this trip have been my favorite for sure. Octopus, squid.
Cool. I think I have another one for you, but let's first see, because we see some comments that people really like the midwater octopus, the Japetella, and also the squids.
Oh yeah, yeah.
So yeah, indeed, we do agree. And some of the baby octopus, and what I was going to tell you, Alex, is that also seeing the skate eggs at the very first time was
That's true.
that was amazing.
That's true.
Too.
I think for like a favorite moment, a singular moment, probably the skate eggs. Finding the skate nursery was super cool.
Yeah.
Overall, I've liked the cephalopods. But
Yeah, I don't know.
Oh.
Looks like it had a huge meal, or it's pregnant.
yeah, is that being gravid or is that just stuffed with food?
Yeah. So yeah, many comments here. So let's try to. Yeah, well, a lot of people saying that the newly hatched octopus, baby octopus, definitely has been a great highlight. Also, the shrimp predating on the baby octopus. Well, let's see what else. So yeah, another comment on the midwater octopus, the Japetella, which is definitely totally cool.
The Dumbo.
The Dumbo was
Yeah.
great. Yeah.
Dumbo was good.
What about you?
No one's saying whale skull. I think that's my favorite.
Yeah, yeah.
Whale
Especially
skulls are cool.
because we are wondering now if it is from an extinct animal or something at least that is not known to be extant.
Yeah.
Okay.
It's so small.
Yeah, it's
Yeah.
so
Yeah.
small. And
Yeah,
as
that-
evolutionary biologists, we have to rely on things like fossils to help us date
Yeah.
our phylogenies. So findings like this are often incredibly important.
Yeah. Whale school was great. Also, I remember, I think it was second dive, we saw a shark tooth.
Oh, on the zoom in?
Yeah, I
Yeah,
remember.
I think
Yeah.
I recall that.
Yeah.
Lots of little pteropod shells on the sediment have been cool. Different from the shells I'm used to. I'm used to like the spiral, very much a flap and swim and snail sort of pteropod shell. So there's been some cool ones here.
Yeah. Oh, also the flatworm yesterday was incredible.
Oh, the weird little pancake flying saucer
Yeah.
thing? Yeah.
What about you guys? Tyler and Gerald here. What have been your favorite moments?
The one of the baby octopus hatching and then the shrimp getting it and then it getting away.
Yeah.
Yeah.
Okay.
Yeah.
Yeah.
Crazy, like.
Okay. So another one for the baby octopus involved.
Yeah.
What about you, Tyler?
Dumbo octopus?
Dumbo?
Dumbo
Yeah.
octopus.
I was flying and watched it.
Yeah. Okay. Cool.
That's true too, right? Because Tyler's coming from this where like this is his first expedition where he's piloting now. And so that event, the Dumbo octopus for you is like, one, it's just a cool event. But two, like you were piloting to try and keep tabs on this animal and yeah.
Yeah,
A learning experience.
totally. So yeah, well, and we do have many comments. So yeah, a lot of people talking about the baby octopus. So I think those are the overall winners of the expedition.
Yeah.
I guess no surprise, but also we see a lot of people talking about even the Siloplax we saw earlier on the woodlock. So yeah, that's pretty cool, and we definitely believe that that might be something really, really interesting coming out of this expedition. So we have been like rushing, getting in contact with experts, yeah, from that are on land, trying to see, yeah, what would be the best way possible. What's that? Swamp lobster. So yeah, keep sharing your thoughts on your favorite your favorite observations and highlights of this expedition, because yeah, this is our last dive. So
Last dive.
yeah,
Sea pig.
sea pig. Oh,
Right there.
oh yeah, can we do a zoom on that? Yeah, we're missing one actually. This Cotoplanis. So this might be our moment.
Sergio immediately goes to the list. Is this one we need?
Oh.
While we try to ID this, if you are maybe in need of refreshing on some of the incredible moments during this expedition, you can, of course, check out all the past dives. Those are on our YouTube. But there's also highlight videos, weekly update videos that are put together. Lots of great enthusiasm this expedition, and just fun moments. And of course, you can also go on Twitter and check out the hashtag Octo Odyssey. Lots of people there were sharing moments throughout the expedition, either screen grabs or video clips, or just kind of sharing their thoughts. So lots of good ways to catch up on this expedition, even though we're on the last dive of part 1 before we come back in December.
That looks way different than the ones we got, huh?
It's got like the two. I mean, it's definitely not Amphirema because it only has. It doesn't have the connective.
Peniagony because it has just like one.
Yeah.
All right, can we get it?
Is this one of the ones you were looking for then, Sergio?
Yeah, I mean, actually it looks very different to the ones that we were looking for.
Yeah. But it's one we haven't seen then yet? Or
Yeah, not
Okay.
very similar to
Similar.
the one we got like 2 dives ago.
Yeah.
But yeah, the upper papilla are looking different. So it looks like it's like bifurcated. You see?
Yeah.
And it only has the two.
Yeah. The other one was just like one. So we wanted the Amperma.
They've
But
been
yeah.
surviving well. I mean, intact specimens in the sections that were
Mm.
Yeah, we might try.
I defer to Jay or Adam on that, but.
Yeah.
Well, I was getting a suction, so I just assumed that's what y'all wanted.
Okay.
Suction?
Uh-huh.
Yeah, thanks.
But I
Yeah.
can do whatever.
No,
Let's do it.
that's what I was thinking.
I mean,
That's
there
it.
was the psycho that got put in the bio box.
The what?
The y'all's big one
Yeah.
that went in the bio
The
box.
Yeah,
psychopodies.
The psychopodies.
Which
Yeah,
is
the-
actually twice as large as the whale skull.
Yeah.
Crazy.
If
Totally.
y'all want to see the enthusiasm that the scientists have, go scroll through that hashtag Octo Odyssey and find the photo of Holly and Sergio with this sea cucumber that they collected. First of all, just a massive thing. It's difficult a lot of times judging the size, which is why we have these lasers to tell us what 10 centimeters is. I never would have guessed that sea cucumber was going to be that big. So check out that photo.
Yeah, it was massive.
On Twitter.
Yeah, you can find it on Twitter. Yep. And well, yeah, probably the record just on Twitter. I guess it was shared over Instagram, but just in stories. So yeah, head over to Twitter and you can see the pictures.
Is this going into which
Which chamber is it going? 6.
six? Okay.
So we're gonna try to sample this type of sea cucumber, holothuroid, and just we had this conversation. I think it was in our previous dive. there are some names for this type of sea cucumbers. For example, like sea bunnies, or sea pigs, or gummy squirrels. You know, many different names. But definitely, one thing is that they are all sea cucumbers, right? And then you have this different names for different categories. I'm not gonna go into detail on each one of those, but just for you to have as a generality that all of them are sea cucumbers, so holothurians. And these guys are called the engineers of the seafloor because, as you can see, of the in their you can see their intestines, their guts. They consume a lot of sediments, so they are deposit feeders, and they're fixing a lot of carbon here.
Cilia on it.
Yeah. So you annotated that?
Yeah, I will. I keep.
There it is.
Okay, cool.
I'll add that as a comment.
We're all just quiet, waiting for it to come up into the suction sampler cylinders. There
Okay,
we go.
there
got
we
some
go.
sediment.
Nice.
There it is.
Thank you.
Sorry,
guys.
we're all watching another screen. So I'd kind of explained how the suction sampler worked before, but we actually have a little rotating tray of cylinders that we can spin around. We have another little grainy camera here that we can see what number and whether there's something in that cylinder. It's not fantastic footage, but whenever they suction this up, it of course has to make its way all the way through the suction tube before it gets to that cylinder. So we all kind of just wait with bated breath for it to show up there.
Yeah. So we just finished to getting that holothurian sample, and we're gonna keep exploring around to see what we can find here. Y quería comentarles a los que nos están viendo en español un poco de lo que hemos estado hablando. Bueno, primeramente invitarles, así como les decíamos en inglés, estábamos haciendo una recapitulación de los momentos favoritos que hemos visto en nuestros buceos. Entonces, por ejemplo, yo les estaba comentando de la quimera que vimos ayer, que también se le llama tiburón fantasma. Y bueno, por aquí varias personas en el cuarto de control y en el chat están comentando que sus momentos favoritos han sido por ejemplo, ver los pulpos bebés, el momento de los nacimientos. Así que si ustedes nos están escuchando en este momento en vivo, por favor déjenos saber cuáles han sido sus momentos preferidos, sus, por decirlo así, sus observaciones estrella, y nos lo pueden dejar en los chats de Facebook o de YouTube, y por acá los estaremos leyendo. Y también pues al mismo tiempo comentarles de que muchos de estos organismos, por ejemplo como el que veíamos anteriormente, son pepinos de mar y tienen diferentes nombres comunes. Algunos le llaman conejos de mar, otros le llaman cerdos de mar, algunos se le llaman ardillas de mar, como ardillas gomita, pero todos son nombres comunes para diferentes tipos de pepinos, pero al final lo que comparten en común es que todos son pepinos. Y que de nuevo, para los que no lo tienen claro, son del grupo de los holoturios o holoturoideos, que son parte de un grupo mayor que son los equinodermos, o sea, el grupo de las estrellas de mar, los pepinos de mar, los estrellas quebradizas o fiuras y los crinoideos. Y bueno, pues justamente hoy vimos un animal en particular o una especie muy enigmática en un pedazo de madera que vimos en la mañana, y que digo enigmático porque todavía es un debate a nivel científico taxonómico si son un grupo aparte de los equinodermos o si son parte de los asteroideos, o sea, de las estrellas de mar. Así que, de nuevo, les invito a que si tienen alguna pregunta, comentario, que nos la dejen en los chats, pero que al mismo tiempo, si quieren compartir cuáles han sido sus momentos favoritos de esta expedición, que nos lo pongan en el chat y por acá los vamos a estar leyendo. Y para nosotros pues es de gran importancia también saber ¿Qué han pensado ustedes que han estado siguiendo estas transmisiones durante estos últimos 20 días?
Pretty long. I don't know exactly.
We don't have an exact number on how long that suction hose is, but if forced to guess, J-Rod will say about 2.5 meters.
So we got some reports from people from Team Cuke. People like
Yeah.
us who love sea cucumber. So yeah, we're I guess Team Cuke is here in the house right now in the control room. So yeah, woohoo! We love sea cucumbers.
It's very important, right? Like you mentioned, engineers of the sea, they do play an extremely important environmental role.
Totally.
Yeah.
Yeah, and literally just by pooping, eating and pooping. So I don't know if I'm allowed to say that, but
Yeah.
defecation.
Of course you can. Yeah. All right, settling back down. I'm going to pull up a map here real quick. It's been a hot minute.
Y veo una pregunta en el chat de Facebook mientras nos vamos moviendo al siguiente punto. Si estamos en el fondo del mar, y pues sí, estamos ahorita a 3029 metros de profundidad. Estamos, bueno, Alex nos está haciendo el favor de mostrar en el mapa dónde es que estamos. Entonces, ese cuadrito que ven como en amarillo, pues es donde estamos justamente ahorita. Y entonces sí. Así que si en algún momento ven que estamos viendo la columna de agua, no piensen que estamos ascendiendo, sino es que estamos reposicionándonos. Wow, mientras vemos un pulpo. So, yeah, special thanks to Alex showing us the map of where we are right now while we see this beautiful octopus. And in this northern region of the Dorado Outcrop. Everybody. Yeah. So what are those?
I think it's just sponge. I
Yeah.
thought they were like saggy eyes at first.
Look like, right?
There's sponge, and it looks like
Sponge.
a sea dandelion right under them.
Yeah. Oh yeah. Uh-huh. So really nice octopus here. Probably moose octopus. Very similar to the one we have seen in this area. Yeah. Looks like, well, let me have a closer look at the... I guess that's a male, right? But
We haven't seen a clear view of the
the
third right arm yet.
hectocotylus, yeah.
Yeah.
We could pan around to its right side.
Yeah.
So for
It's,
those of you who wonder, if we pay special attention to the arms of this octopus, looks like a female, yeah, and that's because in one of the arms they have a specific structure, the males, to transfer the sperm to the female. So looks like this one is actually a female because it doesn't have that hectocotylus. That's how the name, technical name of that structure.
Yeah, and that's the modified third right arm on this particular species. Will be shorter and have a grooved tip to it instead of having sucker discs at the end of it. And that is used by the male to pass spermatophores to the female. There are some cephalopods where the hectocotylus will come off, but not for these ones, not moose octopus. And honestly, a lot of octopus, it's just used to hand over a sperm packet, and that's not to say that they're only reproducing once. So something like a giant Pacific octopus is another one that you might see a lot of at an aquarium, where a male has a distinct third right arm.
we've seen a lot of circular scars on the male host octopus, but not really seeing any on her.
That's a good point. The males have been really scarred up. And the female here, I mean, she's got some marks, but not nearly like the old scars that we keep seeing on the males. Yeah.
Estamos viendo una de las especies comunes de pulpos en esta área, muy probablemente el género Modus Octopus. Y estábamos justamente hablando aquí con los colegas en el cuarto de control de que este parece ser una hembra. Y esto es debido a que no le observamos una estructura específica que los machos tienen en su tercer brazo, una estructura modificada en su tercer brazo que se le llama ectocótilo y que básicamente es para transferir, sus paquetes de gametos o espermatóforos a las hembras. Entonces no lo vemos en este espécimen, entonces parece por esa conclusión de que es una, llegamos a la conclusión, de que es una hembra.
So octopus are terminal breeders. They breed, or they really go into a single reproductive phase at the end of their life and become senescent, where they stop eating. The males at that point kind of just roam around looking for mates. The females, after mating, a couple times possibly, will actually then settle down and brood the eggs. But at that point, both animals are kind of just towards the end of their life, and that senescent phase just leads to them sort of growing weaker as they're not eating, and they're solely focused on that reproduction.
Yeah. Oh, he's trying to. Oh, he's getting.
I kind of want to watch what she does with all the
Yeah. Can we just remain steady and just? Yeah. Yeah. We're just gonna remain here.
maybe suggest why they like this cleaner rocks a bit more.
Yeah. Something I've noticed that those sucker discs are chemoreceptive. So they basically taste everything that they're touching.
Yeah, let's leave it to.
Could be a bit of an odd experience
So
to
maybe-
touch a little siphonophore like that.
Yeah, just keep the camera steady. Yeah.
After it gets out.
Okay, cool.
So we're just gonna sit here and let her wander.
I'm about to text her.
Nice. Actually, yes, she's.
Yeah, there's another one on the right.
Okay, so yeah, let's wait until the octopus gets out, right? And do a close-up of the daylight. Okay.
Okay, Nat says not to worry about the screen exit, and she would like to switch to the close size.
Okay.
Abrahams on it. Oh, he said she's gotta be zoomed in for it to work at her walking speed. So maybe we can get that now.
Yeah. Oh, really nice perspective.
Yeah. Look at how much she's getting up.
Yeah, so for the ones who are wondering if she had any eggs, doesn't look like it, and we didn't see any eggs nearby, so this might be probably just a wandering female that hasn't uh, laid eggs yet.
You do see a little bit of the circular scar.
A bit, but it's not like on those males.
Right.
Yeah.
Theirs look like they're just so old to heal. Those eyes are beautiful. I'm also not seeing parasites on her.
No.
And we've seen a lot more of those on the male.
Okay, so can we try a shot, guys? So can we try to give a little zoom out and just like leave her in the?
Wait.
We're in the middle of.
Yeah, they're doing that. Ask for us to hold this one.
But I'm talking to not here.
Oh, you are also.
Yeah,
Oh,
yeah,
okay.
yeah, yeah.
So many.
Sorry.
Yeah, sorry. Yeah. No worries. Yeah, I confirm with her.
Done with this?
Yeah. She's happy with this. So. Corner, and as she's like given the feeling that she's climbing up, right? So like that.
There we go. We're all just sitting in here watching her make her way across the land.
Videos de estos pulpos y esta pulpa, particularmente es una hembra, porque no solamente estamos recolectando observaciones para fines científicos, sino que también para fines de comunicación y luego poder poner esto en materiales que nos puedan servir para comunicar a otras personas la importancia de estos animales y esos ecosistemas de aguas profundas.
Can you ask Nat if she wants any tight zooms before she gets too far away, given we can reposition, but is she gonna want anything tight on any tentacles like before?
Yeah. She's afraid. Alright, so she says to wait to get her to
After that.
after that, yeah, and
I just asked here if we had an idea for the lifespan of these animals, and no, there's a lot we don't know about these. Some octopus are very short-lived. Some are a little longer, like a giant Pacific octopus is about 4 to 5 years. We don't know. We don't know how long this one lives.
That should be pretty good for the landings on the plan as well.
this one would definitely be in the multitude of years here. Yeah. But how many years we don't know.
Yeah.
15 to 20?
Ooh, estimate of 15 to 20 there from Janet. And is that going off of, right, there's this idea that the brooding period of these animals is roughly a quarter of their lifespan. And so observationally, they've seen them brood for a long enough period of time that if that was just a quarter of their life, their whole life would be about 15 to 20 years. Yeah.
If the metabolic rate is the same in this group as in granulated.
Okay. Yeah. It all depends on the metabolics. And then, of course, that changes when you're living at a vent and it's warm water.
Right?
Yeah.
If you're in ectotherm, you take up the temperature that the environment has. And that may be why these guys like to be warm. Although notably, the only species of octopod we've seen in these shimmering fluid flows is this one.
Even though we have seen other octopus in the area, they don't seem to be using these flows.
No.
Yeah.
So Janet, you said between 10 to 15 years, this one.
Maybe.
Maybe. Okay, I'm just gonna
Yes.
say it in Spanish.
It's not 2 or 3.
Okay.
These are, I mean, big octopus.
Yeah. Acá la especialista en cefalópodos, la doctora Janet Boyd, nos está comentando de que sus estimaciones, ¿verdad? Ella como experta que ha trabajado en este tema durante muchísimos años, ella estima que esta pulpa puede tener entre 10 a 15 años por su gran tamaño. Difícil saber, ¿verdad? La edad exacta, pero tenemos ese estimado de que pues es un animal pues que ya ha pasado su más de una década al menos viviendo en estas aguas profundas.
Likely, okay.
No, I know there's stars, but they're not like those white circle stars that we have at the bottom. I don't see the circle. Like no, I didn't. Is that the female? I'm saying that the males have.
Yeah, it's a female.
Like the ton, like this all over her head. She just has one. I just was wondering if you were seeing that on the other female.
Y bueno,
Just observing and asking.
voy a hacer énfasis aquí en una pregunta que tenemos de una muy buena colega de la Universidad Nacional, Sileni, que pregunta si estos pulpos tienen cromatóforos o si lo usan a estas profundidades. Y efectivamente sí, pero no los utilizan tanto como los utilizan, por ejemplo, los cefalópodos de aguas más someras. Entonces, por ejemplo, a veces hemos visto que dependiendo del ángulo que le pegue la luz del ROV, a veces varía un poco su tono, y eso era lo que nos comentaba la experta Janet, de que a veces parecen de que tienen diferentes colores, pero que mucho es por ese ángulo de incidencia de la luz. Pero a diferencia de otros cefalópodos, y eso lo hemos estado hablando muchísimo durante el crucero, no los utilizan tan activamente, puesto que aquí pues no hay tanto, ¿verdad? Bueno, primero no hay luz en general, pues casi que es oscuridad total, excepto por la bioluminiscencia de los organismos que viven acá. Entonces no hay tanta necesidad para ese tipo de mecanismo. Así que pura vida, Sileni, por la pregunta. Y bueno, pues con esto aprovecho para despedirme y decirles que para mí ha sido un placer, de verdad, acompañarlos durante todos estos buceos. Pues me voy a poner un poco emocional porque sí que es un momento muy especial, porque este ya es el último buceo, lo que quiere decir que es la última vez que los voy a estar acompañando en esta transmisión. Pero espero estarlos acompañando de nuevo en diciembre, si todo sale bien. Así que le voy a pasar el control del buceo, el liderazgo, a la doctora Odalisca Brady de la Universidad de Costa Rica, que es experta en octocorales, y ella les va a estar acompañando por la siguiente hora. Así que todavía nos quedan algunas horas de buceo, unas 2 horas de buceo más. Y pues los invito a todos y a todas a que sigan conectados y disfrutando de esta transmisión. Para mí de verdad ha sido un placer y un honor estar aquí en representación de muchísimos ticos y ticas que son apasionados del océano y exploradores de las aguas profundas. So thank you everyone. I'm going to pass it over to my colleague Dr. Odalis Cabriti from Universidad de Costa Rica. She's an octocoral expert, and just would like to thank you all for joining us in this amazing dives and also sharing your favorite highlights and moments. For me, it has been a pleasure to be with you during all these dives, and it's a kind of a nostalgic moment for me because this is the last dive, but I'm sure we will have more dives in December. So special thanks to everyone, and I'm gonna hand it over to Dr. Odalis Cabriti. Thanks and pura vida. Yeah, nada, it's like losing it. De solo explorando.
¿Pero hay alguna ruta?
No, solo quedarnos por aquí buscando las cosas interesantes. Sí. Digamos, nosotros estábamos tratando de aprovechar para hacer las tomitas de Natalie, así como de bonitas. Entonces también si usted ve, por ejemplo, algún octopus que sea bien bonito.
Para hacer tomas.
Sí, para hacer tomitas y igual de los pulpos.
¿Qué fue lo que Jorge te pidió que hiciera?
Quisiéramos de los dandelions, de los sea dandelions, entonces que están por ahí abajo. Entonces sí, igual lo va a decir a Jerry. Just you guys over here from the sea dandelions. Don't remember what they are.
Hey, hello everybody. Thank you for being here, joining us in this interesting exploration. This is dive 542 of our ROV SuBastian on the Falcor 2 ship boat. And this is our last hours of dive. We will be diving until 7 maybe. And this is close to the end of the expedition that has been very successful. We have a lot of interesting results that will be analyzed in the future. In future months, we will be analyzing data that was collected here and also samples that are taken from this expedition. And since they had experiments, some of the researchers had experiments here in this area before, they will be comparing the data from 10 years ago against the data that they collected from this expedition. This will take some time, and then you will see the scientific publications of their findings in the future. I'm here with the experienced pilot of the ROV, with Tyler, J-Rod, and Alex in the media, and also with Holly, who is a microbiologist, and she's going to be with us also. She also studies octopus. And we have here Janet, who is an expert in octopuses that you heard some minutes ago, that she was explaining about the age of the octopus and some of the behavior that they have. Our idea now is to take these beautiful shots in the last 2 hours that we have of the dive. Hola a todos, que gracias por estar con nosotros en estas últimas horas de expedición. Ya nos quedan solo como 2 horas, ya la expedición se termina después de 22 días de trabajar en estas zonas. Aquí estoy acompañada por los expertos pilotos del robot SuBastian. Entre ellos son Tyler, J-Rod, y tenemos a Alex en el multimedia. Ahí en este momento estamos viendo unos briozoarios coloniales que son ramificados, esas ramitas blancas que ven ahí. Les decía que en estos momentos estamos haciendo algunas tomas de los pulpos porque además de recolectar muestras, lo que se hace luego es analizar videos. Todos estos videos van a ser analizados y este trabajo es un trabajo de horas de estar revisándolos para darse una idea como de todo el ambiente marino, de la abundancia de los organismos que hay ahí y muy importante, es la única forma de poder entender el comportamiento de un animal. Por ejemplo, las horas de video que hemos tomado de este pulpo van a hacer que Janet y Holly analicen todo esto y vean un poco y aprendan un poco más sobre el comportamiento de estos animales. Entonces, por ahora les digo que disfruten de estos últimos, digamos, estas últimas 2 horas de video que vamos a estar proyectando. Y bueno, a ponerle atención a algunas de estas cosas que probablemente no veamos por mucho tiempo. En este momento también estamos buceando en el monte El Dorado, que es donde empezamos la expedición, que fue como el punto clave que se tenía para encontrar estos lugares donde los pulpos anidan. Sin embargo, en esta expedición encontramos que también anidan en otros sitios y eso es un hallazgo muy importante para comunicar. We are in the Dorado Outcrop, the site where we started this expedition, and it's the site that we thought that were the only site where we can find the octopus, the Musoctopus, this genus, and he was brooding here. But we found in this expedition other sites of other nursery sites, and this is a very important finding that we have from this expedition. Now we are taking some video of the sessile organisms. Ese es un organismo que le dicen diente de león, es un hidrosuario. No, it's not an hydro. Yeah. Un nidario del grupo de los nidarios. Vean esas estructuras como parece como una tela de araña, que son extensiones del organismo. Vean qué colores más bonitos tienen. Y en el fondo lo que vemos son sedimentos, y lo que vemos como una lluvia que está cayendo son sedimentos que caen de la parte superior, sedimentos que caen de la columna de agua.
sometimes I like that.
Como les digo, una vez que se tienen, digamos, esos organismos que se han recolectado en alguna expedición, no es posible ver este tipo de estructuras que tienen y poder hacer hipótesis sobre qué son y para qué les sirve. Nothing to collect for me. Oh, I think we already collected most of this. The octocorals in this area, I already have some. So, yes, did you find something new in the last 2 hours?
You guys need any more footage?
I don't know. I think it's okay. We can go on. No?
Yeah, I think whatever.
Yeah, we can continue.
Yeah.
Yeah,
Want to keep going?
yeah.
Find something new?
What?
Find something new for you? We can?
We just go there and take more video. We will see if something interesting appears.
We still have a lot of space. So
Yeah,
Last time.
they are free, but I don't see the point in collecting things that we really collect.
oh yeah, true.
But if I find something interesting, we have a space.
Yeah, I'm
It's
just
okay.
making sure you know. Yeah.
Yeah, yeah. Okay. And if you see something that you want,
Mhm.
you let me know.
Okay. Yeah, I'm happy with the sea star and the sea cucumber.
Okay.
So.
I saw that you took the sea star.
Yeah.
Okay.
Look like they're smiling.
And we have seen this many times during the dive. I think it's a bryozoan. Is it difficult to catch? Maybe this thing. We haven't collected, but we have seen it many times during the dives.
You wanna get close to it?
Maybe it could be interesting to take a look and see if we can take a piece or something. Sponge. Here, this thing. This one. Yeah, that's one. That one.
The same picture. What your lures on for London, you can see it with that.
Yeah, it seems like that. Yeah.
So bring your first side.
She wants to.
Oh, this one?
Just to take a look of the specimen. Eso que estamos viendo parece que es un briozoario, lo hemos visto muchas veces en estos buceos. Vamos a verlo un poco de cerca, a ver si vale la pena pues estudiarlo o si no. It looks that it's covered with sediments, huh? Yeah, and it's full of other organisms here. You already had this shot, huh? I think that we saw that before. But I think that, I don't know, maybe like here? Is it possible to come here? Mm-hmm.
Or pushing ahead is the one to do. Rather than look down first.
this is interesting.
Yeah, so just come in nice and slow, and then just ease your forwards, and you still need a little bit of downtrip unless you catch underneath the ledge. Yeah, to sort of use the buoyancy, keep you steady, but you get a bit high for this. You can get this bit. Yeah, try it there. Keep it steady. Get happy.
Sí, esos son colonias de briozoarios muy cubiertos con sedimento, entonces sí, tal vez no vale la pena sacar una muestra ahí. This looks beautiful. Can focus like here. Yeah. Yes, okay. You can see the structure of the bryozoan there. It's another colonial animal, very interesting. A lot of them in the fossil record. It's a very old group of animals. Ahí vemos ese briozoario que es como ramificado, tiene como una malla, está formado como de unas diferentes hojas, como una malla. Y los briozoarios son muy antiguos en la historia evolutiva. Este está todo lleno de sedimentos. This is full of sediments. Ahora nos vamos a seguir moviendo aquí en sobre este monte para ver si encontramos otros organismos interesantes. Bueno, todos son interesantes, pero alguna muestra de las que tenemos como objetivo. Y también, obviamente, para hacer más video. Ahí pueden ver cómo cae ese sedimento, todo ese sedimento que está en la columna de agua que va cayendo sobre el sustrato. Y forma a veces unas capas muy grandes y como vimos en ese briozoario, a veces cubre totalmente los organismos. Y por supuesto provee un ambiente apropiado para muchos otros, digamos, organismos sésiles que ese ambiente como, digamos, lleno de sedimentos les favorece mucho. Y muchos de estos son filtradores, o sea, se alimentan de organismos en esa columna de agua, en esos sedimentos que van cayendo.
Try and get more light for your better. So you can see it.
Yeah, it's
Maybe that or a bit less.
a little more.
A little bit more.
Actually, go down one again. Sorry.
And one.
Yeah. Down one more. Yeah.
And a little bit of light. So not that.
Slow motion over there now.
That's really
Ahí tenemos una anémona bastante grande. Can you go closer to that sponge? This one, just to take a look.
Big guy?
Sponge.
No, take it, wait.
Ahí vemos unas esponjas que son de los principales habitantes de estos fondos submarinos. Wow. Yeah. Can you put the lasers in some moment? Okay. 10, 10, 10. I think this is kind of hard, but maybe you can break a little piece of that. Yeah. Okay. Like around here, that looks not so difficult because I don't dare to say easy because it is not. Ya vamos bueno, ya ahora vamos a tomar un video más cerca de esa esponja y la idea es como poder recolectar un pedazo, pero vamos a ver porque estas esponjas son muy duras. Estamos viendo estas esponjas. We are seeing some sponge here, and this sponge is very hard. So we will try to see if it is possible to collect a little piece for study. This type of sponges form very huge structures, and they are important constructors of the 3D relief of the bottom of the ocean. And as you see there are some crinoids on it. So there are many organisms that live associated to these sponges and also corals. And in the left of the sponge, you can observe some other types of bryozoans, one crustacean and one coral, one hard coral. And if you look with pay attention to the bottom, you will see some polyplacophorus and a lot of organisms. Si ponen atención, ahí van a ver algunos crustáceos en el fondo, unos briozoarios en la parte izquierda, más arriba un molusco y otros tipos de esponjas alrededor. Okay, we have a change here. Now we have Carl and Tyler finish his shift. Okay. Thank you, Tyler. Ahora tenemos a Carl, que está de piloto del ROV, del robot, y ya Tyler terminó su turno. Como yo les he dicho en otras ocasiones, ellos están cambiando constantemente porque es muy cansado lo que ellos hacen y tienen que poner mucha atención. O sea, tienen en sus manos un instrumento, digamos, un juguete demasiado caro. Entonces ellos tienen que tener mucho cuidado, tienen que manejar muchas variables para poder trabajar aquí, poder recolectar una muestra, tomar buenos videos, manejar las cámaras y además cuidar el aparato que no se vaya a dañar. Wow, that's so beautiful. Este es un acercamiento muy lindo de esa esponja. Ahí se ven las fibras, son unas fibras como de vidrio, son de sílice. Esa es una característica de este tipo de esponja, por eso son muy duras. Y incluso cuando uno las toca se puede hasta cortar. So you have a close-up very nice of this sponge. They are made of silice. They are strong, very, very strong, and when you touch them, you can cut yourself because you're seeing there a crinoid who lives on the sponge. You can see that. And I was saying that here the pilots have the responsibility to take care of the ROV, not only to maneuver it, but also they have to take care of not damaging the equipment. They have a very expensive equipment in their hands, so they have to be very careful. And that's why they change frequently. One pilot substitute the other one because they get tired of paying so much attention to the operation. Vamos a tratar de recolectar ahora un pedacito. Ahí pueden ver los láseres, los dos puntitos verdes que son 10 centímetros para que se den una idea del tamaño de esa esponja. Puede tener un medio metro de lado a lado, tal vez más. Y ahora poniendo los láseres podemos ver como qué tamaño de muestra podemos recolectar y que pueda caber dentro del quiver que tenemos que es como un tubo, que es donde metemos las muestras. Esos tubos que tienen una tapa tienen cierta profundidad, más o menos 30 centímetros. Entonces siempre hay que calcular que la muestra pueda entrar ahí. You can see the lasers, the two green dots. They are 10 centimeters, so so it is possible to have an idea of the size of the specimen or the size of the animal when it is alive, and the pilots can calculate if they can take a piece that fits inside the quivers, because the quivers have a limited space. They are not very big. So if they catch something very big, they cannot put it inside the quiver. So with the lasers, they can calculate the size of the piece that they are going to collect. Cuando ellos recolectan una muestra, uno de ellos tiene que mantener el ROV estable mientras el otro está manipulando el brazo para poder tomar la muestra. O sea, es una operación muy coordinada entre ellos dos, entre los dos pilotos. Aquí alrededor vemos muchos otros organismos. Ahí se ven unos gusanos, unos camarones. Bueno, más bien los tubos de los gusanos. Otros tipos de esponjas. Ya. Bueno, ya ellos recolectaron la muestra y ahí se vino un crinoideo, que de estos organismos casi no se sabe nada. Es otro de los organismos que son como el objetivo para los estudios taxonómicos. Vean qué bonito.
One more time.
¿Ya los puso? No, los tenía puestos. Ok.
Yeah, I don't know.
Okay.
For now, 'cause you'll have
Okay. Now they are collecting a piece of the sponge with the crinoid there. This is one of our target species. They are very not well known, so it's important to collect some samples to send to the specialists in the group. And now we have the lasers and we can estimate the size of the crinoid. Remember that this is 10 centimeters long.
okay. Thank you. Sorry.
Okay, thank you. Ahora vamos a recolectar esta muestra. Estábamos un momento calculando el tamaño porque esto es muy importante, porque muchos de los animales se encogen cuando se recolectan y entonces es muy bueno después poder tener los videos y poder saber cómo eran cuando estaban vivos. La idea de todas estas exploraciones es que una vez que nosotros hemos recolectado muestras, hemos recolectado esto, lo estudiamos, podemos hacer la coincidencia entre la muestra que nosotros teníamos y el espécimen cuando está vivo digamos ahí, en el campo, in situ. De esta manera llega un momento en que no es necesario recolectar nada y toda la investigación se puede hacer con solo el análisis de videos. Aquí en nuestro caso es la colega Beatriz Naranjo la que se ha dedicado a este asunto del análisis de los videos y a recolectar todas estas imágenes. Y por otro lado tenemos a Wendy Matamoros, que también ella lo que hace es que con los organismos que hemos recolectado, ella hace como una coincidencia entre esos organismos con las fotos submarinas y con las fotos que se le toman cuando salen a la superficie. De esa manera nosotros vamos haciendo un catálogo con todo esto y estudiando los videos, y de esa manera pues en el futuro, cuando se quiere estudiar el fondo del mar, ya se va a tener una idea clara de qué son los organismos. Nada más va a hacer falta pues recolectar algunos que sean nuevos, que no se hayan estudiado anteriormente. En nuestro caso, nada de esto se conoce, entonces por eso es que se recolecta para hacer estos análisis. In this case, we are collecting this piece of the sponge with the crinoid, and I was telling in Spanish that the idea is that we can match the submarine picture, the in situ animal, with the piece that we collected. So we have people who are researchers who are doing this type of match with the pictures, with the animal when it gets outside, with the living animal, and with the analysis of the morphological structures in order to have the species. And at the end, the idea is to have a catalog with for example, with this sponge in situ, alive, with the sponge collected, and with the sponge with the morphological characteristics. For example, the sponges have spicules, and these spicules give you an idea of the species of the sponges. They are very difficult to study. Also, the taxonomists of sponges, they rely a lot in the molecular studies. Good, thank you. So now we have another sample. Bueno, entonces ahora ya tapamos el recipiente en que está la muestra y ahora procedemos a ir a otros sitios. Sí, una vez cuando, a la hora en que recuperamos el ROV, cuando sube, nosotros estamos en la superficie, todos los investigadores, esperando que lleguen las muestras y preparamos agua de mar fría en bandejas y entonces cuando llegan nosotros recogemos los organismos, los ponemos en esas bandejas, todos con guantes, ¿verdad? Para no contaminar las muestras y las ponemos en una cámara fría donde se mantienen a la temperatura en que están en el fondo del mar. Bueno, no tenemos así como que esté a 1 grado centígrado, tenemos algunas de esas son más frías, pero es como vamos a tener la muestra. De ahí la sacamos y se le toman fotografías a ese espécimen, así recién recolectado, y después se pasan al laboratorio para realizar las submuestras y fijarlas. Las submuestras son para análisis de ADN, para estudios de microbioma, para lo que se necesite y se trabajan con diferentes protocolos. Al final, la parte de la muestra que queda se fija, ya sea en formalina o en alcohol, para que sea trasladada al Museo de Zoología de la Universidad de Costa Rica, donde esas muestras van a quedar depositadas y donde se van a estudiar y también se van a tener a disposición de científicos extranjeros para que puedan llegar a nuestro museo o pedir las muestras en préstamo, que es una forma en que se hace para compartir muestras con especialistas que no hay en el país, digamos. Aquí tenemos especialistas, pero obviamente no en todo. This yellow sponge is beautiful. Mm-hmm. Looks like it looks like dry or something. Yes, I think so. So it's weird. Mm-hmm. Yes, it's covered in sediment. Wow. There's a sponge covered in sediment in or on. It's covered in. And there is another sponge, the yellow one, that is very, very beautiful. So in this place we have a lot of sponges, a lot of types of sponges. Este es un sitio en que tenemos muchos tipos de esponjas, esas grandes que llaman la atención como esta amarilla, pero también hay unas pequeñitas que están pegadas en las rocas. It seems like a dead sponge, maybe. Ahí, por ejemplo, estamos viendo como una esponja que seguro se despegó y bueno, se cubrió de sedimento. Estos animales que viven en estas profundidades en general tienen unas vidas muy largas. Las condiciones son más o menos estables y cuando ellos mueren es porque se arrancan. Se arranca por alguna razón, porque bueno, algún animal lo arrancó o por una corriente o por alguna razón que disturbe el fondo, entonces ellos se pueden romper. Y bueno, pues son miles de años en casos de algunos organismos que simplemente se arrancan y ya desaparecen. Imagínense lo que podría hacer aquí, lo que pasaría aquí si se hicieran, por ejemplo, un arrastre con una red, porque arrastraríamos una cantidad de fauna que va a durar miles de años para recuperarse o no se va a recuperar nunca. Entonces es algo como que hay que pensar, ¿verdad? En todo lo que es la exploración, la explotación del mar profundo es algo que tenemos que tener muy en cuenta, que tenemos que tener presente y que tenemos que tener cuidado porque ahora hay muchas iniciativas de hacer eso y vamos a destruir nuestro patrimonio submarino, que es, como hemos hablado, el 92% de nuestro país, es un país submarino. Si quieren hacer preguntas, pueden hacerlas en español, portugués o francés, que son inglés por supuesto, que son los idiomas que manejan los científicos que estamos en esta expedición. Some organisms, we can find some organisms that are disattached when they get loose and they fell, they die because they get covered on sediments. And we don't know why they get disattached, mostly maybe because an animal attacked them or just hit them, or because of some conditions of the currents or different things that we don't know in this moment. But as you can see, these animals are very long-lived organisms and they die when they disattach. So imagine if you use some destructive fishing equipment and do like a net over the bottom, on the bottom, so you will kill and break a lot of animals that it will take thousands of years to recover the environment. It is not easy. It is not easy to recover. When we destroy the bottom of the sea, it takes years and years to recover again. And we can see that because the experiments that were set there 10 years ago, they don't even move from where they are. So if you destroy something, it will be destroyed for decades. I mean, thousands of years. So Costa Rica is 92% deep water, ocean water. So we have to think about that when we decide. We have to be aware of that and protect our deep environment. There's a sea cucumber, and the bottom is made of small lavas, small basalts. También vemos ahí un pepino y el fondo son unas rocas, pequeños basaltos y algunas otras como más grandes que se ven ahí. También es importante estudiar la composición de ese fondo oceánico y eso lo está haciendo aquí el grupo de los que trabajan en geología. Están estudiando las rocas y estudiando cómo se han depositado aquí sedimentos, etc. Ahí tenemos un pulpo y también tenemos un pepino cubierto de estrellas. Vamos a hacer algunas observaciones de este pulpo. Here we have another octopus, the moose octopus, and we are going to make some video of him. Him or her? Hembra o macho? Is it a male or female?
I think it's a female, but it's hard. Oh, actually, no, I'm sorry. That looks like the third arm is a female.
It looks like a male?
Yeah, but I can't tell until we see this arm, but Janet's been saying the males hold their right arm tucked in.
Ah, okay.
So we're able to, if it's not difficult to look at its right side, we might get a better look.
Okay.
But no worries.
So we are going to observe the octopus. Como les había dicho antes, estos pulpos del género Musoctopus es un pulpo que es uno de los objetivos de esta investigación. Este es un macho. Gracias, Holly. Este es un macho y vamos a hacer unas tomas de él. Ahí a la izquierda tenemos un coral escleractinio, un coral de copa.
That one looked pretty big. Did you want to see later one?
Oh shoot, you're right. Sorry. Did we get these?
Sí. Mm. Ahí tenemos los puntitos verdes que son los láseres para ver de qué tamaño es el pulpo y vemos como que la parte de la cabeza puede tener tal vez como unos 15 centímetros. Es un pulpo grande. Para este género. Bueno, muy bien. Yo aquí termino mi espacio de hoy y muchas gracias por habernos acompañado estos días. Espero que nos veamos en diciembre en otra expedición al fondo del mar. Ya este es el último buceo y fue un gusto estar con todos ustedes. Siempre nos pueden contactar. Los contactos de nosotros están en la página del Schmidt Ocean Institute y pueden conseguir información, pueden seguir viendo los videos porque van a estar ahí en la página de ellos. Entonces aquí no se acaba esto, es solo el principio. Y sí, muchas gracias. Thank you very much for being with us. This is the end of my shift. I'm going to leave you with Beth Orcott, who is the chief scientist of this expedition. And this is the last dive, the last 2 hours of the dives of this expedition. And it was very, very, very nice to have you these days here, and hope you join us in December. That probably will have another expedition to the deep seas of Costa Rica. So goodbye. Thank you, Carla, for all these days. Hello.
Okay, it's quite a collection. What were our marching orders? Follow this contour? Yeah, there's a saddle behind us. Mike, do you mind zooming out on the map screen just so I can see where we've been? Okay, thanks.
You think follow this one or?
Let's see, that's, which grid is 100? Yeah, probably going over to the other side of it. So once we get to where your hand is, kind of kick over to the eastern face. just looked at the sample log. How you show me the?
Thank you. I just think these were moving in that direction. I think.
Yeah.
Sorry, you guys. Oh,
Mm-hmm. Can we have a moment to look at this, please? Can we get lasers on, please? Thank you. It's very iridescent. Cool. Okay, we can keep going. Thank you. And you can kill the lasers. Thank you. What time do we need to be off bottom? 6:30, 6:45?
Something like that. Yeah, it's about 9,000 meters.
Okay.
It's like 2 hours, 45 minutes.
Okay. 2 hours 45. Okay, so that would be 6:15. Closer to 6:15. Okay.
Sound right?
Sure. Yeah. Can we have another look at this octopus here? I don't think I've seen one with their front tentacle out like that.
Today's been a lot of interesting postures. Movement.
A lot more of these brittle stars on pyrosomes or sponges. Okay, thanks, Jill. We can keep going. Oh, can we pause for a second to see this little black blob? Oh yeah, I think it's that same snail, or something like the snail we saw. Yeah, you can see its little appendages.
They
don't think so.
Well, actually, it's like a dead snail up to the left.
Dead snail to the left.
This little pink.
Oh, I think that's just demo sponge attached to a rock. I can have a quick zoom. Whoop. All right, we can keep going. Seeing some of these psychedelic medusa floating by in the water. Hi.
Hello, how are you?
I'm pretty good. So we're just kind of heading in that direction to get over to the other side, as far as we can get before we need to come off bottom.
Go a little fast.
Sure.
We'll start with us a little.
Southeast.
Yeah, I guess just like bring them over to about like right over here somewhere.
Octopus swimming.
Right ahead of us.
I see it, yeah. It's not moving. No, good. Just wanted to look at this guy.
That'd be great. Got an updated waypoint for you? Updated waypoint.
10 points.
Good lady.
That's a female.
Yeah.
We don't know that.
Do we know anything about their size and age? Probably not.
Apparently said it's not that indicative
Okay.
of final size.
Yeah.
But I know that there's something to do with how rapidly they develop.
Mm-hmm.
So I think she's planning to take closer looks at the juveniles we've collected.
She tricked me. She told me to just do it like that. I was about to take off. I was getting ready to go.
Hey, interesting. She's almost tucking in that third right arm. Wait, maybe it's not.
But it doesn't have the little appendage.
No. They look so wise. They're like elephants.
Mm-hmm.
It's like a nice shot.
Oh, you can like see right through the eye. That's awesome.
Get the laser camera.
Yep. Return them.
Lights coming up. Lasers on.
They've all been just about over 10 to 12 centimeters.
That's the lasers over right there. Coming back in.
Yep.
Or just
Thanks.
more images of her, or keep going?
I think we can keep going. Thanks.
You know, the catfish all the way up. She curved down good.
Yeah, it's basically coming. This is like a little depression between two mounds, so all the water is being funneled right into us.
Sonar up the line, get a minor here. You know. Yeah. 25 times.
The fish seem to like this current. So, if possible, we want to go to the other side of this little ridge.
Oh, I'm sorry.
It's
I thought
okay.
You wanted to come to here.
It's fine. No
So
worries.
Pop up and over?
Yeah. We want to
Oh, I'm
get.
sorry.
Don't worry about it. This is still interesting.
Do we know what these are? Just the anemones?
That's what I would guess. I don't know.
First time I've seen it on my ship. Wait,
1, 2, 3, 4, or it's a cup coral. I'm not sure.
but it's got 8. Wait a minute. That looks like
That's right now you're a bit. I was far away, so as you come in, so it's going to start getting washed out. That's not too bad.
It's pretty cool looking. We can keep going.
I did.
That's interesting. As soon as we come over the rise, you see corals. We haven't seen them on the other side. Just these little baby ones.
Yeah, pull the ship towards me. No, not yet. Not yet. So that's-
How's the current feel up here, Dorado?
It's pushing right into me right now, but it's manageable.
Okay.
It's going to be hard, just to let you know, if I spin around and I'm looking at it,
Yeah.
it's going to want to push me into stuff, which
Okay.
would make me want to have to thrust back.
Yeah, we can maybe just parallel along this
Okay.
and then head to what you thought we were, but just on this side. Oh, there's another barnacle. We saw one.
Did you want to start with
Nope, it's okay. I just spotted it. I hadn't noticed it before until we were on a dive a couple days ago. And there was a whole bunch of them.
There's still a decent amount of current now that it's at the other side.
Yeah? Okay. It looks like
You
it.
can see it, yeah, that's that coming across.
And you can like even see the coral and stuff like moving.
What when is not?
Oh yeah, let's have a look at that octopus. Could be. Can we get a closer zoom on this arm?
Working my way in right now.
Okay.
Get better as I get closer.
Okay.
Tyler, can you just give me a touch screen? That's good for now.
Yeah. And can we get lasers on, please, Tyler?
Parasites all over. Hands missing 2 arms.
I got pop up.
Okay.
Blow it off.
Yep. That's all right.
Oh, it's just got a lot of parasites, like a few of the other males we've seen.
Mm.
We haven't really been seeing that on the females.
Okay.
It looks like it's missing 2 of the
Yeah, it
front
did.
arms, so. So it's been a lot.
We can keep going. We don't need to come back to that
Try
one.
one more time. You know,
That's okay.
real quick. Huh? Keep going or try
Yeah,
one more
yeah.
time? That's what-
We can keep going.
Roger that. That's all. I didn't want to do that. Couldn't have time to find it. What's that?
Oh,
Yeah, there was
I'm
another
taking a
one
look
of
at
them.
it. I just need to like note that it was not this thing. We'll just see like the density after.
Yeah, it'll be interesting to compare to the Annie Hartwell's study where they like put all the little dots where they'd seen it.
Looking in the wind here. Did your screen die?
Yeah, I mean, it's weird. It's there, but it's flat.
Oh, the deburts. The deburts issue. Do you want me to come around these rocks?
What do
I
we-
think I'm almost kind of straddling it, so I'm a little bit more to the left.
Yeah, that would be good. Yeah, kind of head around that way.
Move them to the southeast.
Wow, look at how much current is coming through there. More barnacles.
Where's that current?
I'm sorry, I just saw a barn.
Yeah. Are we live streaming, Josh?
Yeah.
Or is that down?
We're fine, live stream.
Okay,
It's just your screen over there. I just need to
got it.
deal with it.
That's pretty cool. Yeah.
Looks like another.
Oh,
Is that an octopus or is that a
Sponge.
sponge? Yeah.
I'm just gonna have you get this coral, and then I'll come up the hill.
Okay.
A little bit low on the backside right now.
Yeah. It's like a catcher's mitt. Getting all that stuff coming up.
Really can't get this over there without calling me. So I'm working my way up. Sort of like a false slingshot engage.
Oh, there's a cool little jelly in the water.
On this floor over here, slow down just a little bit to let the vessel get ahead of me a little bit more. We're gonna take it speed up just a touch.
Bridge RV, are you able to bring your speed up just a little bit? RV, brake that again. Are you able to, sorry, bring your speed up just a touch? Yeah, no problem. Thank you.
And some really big, oh, it's a big rock, not a coral, off to the left. Getting into more of a coral garden here. There's an octopus at the top of the screen. Some more barnacles on this rock. Can we get the lasers on while we're coming in? Yeah, similar size. Thanks, you can turn them off, Tyler. Can't really tell how it's holding its arm. They seem to fold up both of their third arms. Okay, thanks. Yeah, I think we're good. What time do we need to come off bottom, Jared?
So that's about 6:30.
Okay. So just
For the 9 o'clock.
Yep. Just a few more minutes on the bottom here as we continue to explore Dorado Outcrop. This is the final dive of the Octopus Odyssey Expedition Part 1. We'll be hopefully returning here in December to visit some of these same seamounts again to recover some of our experiments that we've put out to better understand the fluid flow and temperature and chemistry of this environment, as well as to study the microbes that colonize these rocks and some animal settlement experiments to investigate some of the reproductive history of these organisms that we've been seeing. Now I think we're the third shakedown expedition of this brand new vessel. It's been quite impressive. We've really accomplished a lot on this expedition. had a great team of pilots with us and marine techs. Thanks, Josh, and Chris, and Juliana, and Steph. And yeah, we hope you guys can tune in with us in December. Oh, there's another octopus.
I'll try to get down to it.
Yeah, I know you're kind of pulling. I would say that looks like a male, the appendage at the end. Yeah, can we get the lasers? Yeah. Yep. Great. We can turn the lasers off. What's that?
I was just incredible. I've been noting these white circles on the males and not seeing them on the females.
Thanks.
And that's fun.
Did you want to have a look at it anymore? No?
Oh no, we're good. We can keep moving.
You need some out.
Ooh.
No, I don't want it. I'm fine. That's awesome.
No, I feel like I stay with that one, so we can just keep going. Or get to go on enough.
Yeah. That one's totally hiding its arm.
ROV Briggs, do you want me to keep moving south?
Yes, please.
Yes, please. Thank you.
All right. Lasers on quickly, Tyler. Great. Thank you. And you can turn them back off. All right. We keep heading out. J-Rod? Wow, that current is just still coming strong. You can see those corals off to the left just waving. I'm sure Sergio is going to have a fun time with all the current data we're getting on this expedition. Looks like there might be another octopus up ahead, top of the screen. With a potentially a primnoid next to it. I can't quite tell from this distance. Some barnacles on that rock. Hard to tell with that one. It's all bundled up. Okay, you can come wide, J-Rod. That's not a primnoid next to it.
Stay on this side, or could you hop over to the other side?
We're at 6:30, so don't know that we need to really necessarily go anywhere in particular.
Just something to say goodbye on.
Okay.
The one that I'm at the side.
It's a little more sedimented over here, not as many corals. No, it's fine. All the sponges we're going by are like, pick me, pick me. Yeah, I don't. As soon as we came around that little ridge, it got so much more sedimented. I think it's just not their preferred habitat. Okay.
Stop the ship. There's some buckets out. There's RV. That's a good spot. I'm gonna stop there.
Copy that. Also.
Oh, we're getting some more corals again. Maybe that's a sign. Yeah, we're almost back to where we were when you broke away. So hopefully,
Just hoping I can get one at a different angle.
yeah.
Lateral, like crazy. I have to go a little so we could pass.
Oh yeah, there's all the corals up ahead.
How many times I see sponges, I think that
It's a decoy.
really is. Should have just stopped all of those right there, so. I was gonna say.
Yeah, I think this is still in that sedimented area that we were just at. Yeah.
Sorry about that.
It's okay. That's part of the Odyssey.
Yep, we're gonna start your final.
Oh, I don't know that there's an official final, but yeah, sure. Yeah, so thanks everybody for tuning in to the Octopus Odyssey, part 1. We have made some really amazing discoveries along the way. We've had a really great team with us, bringing all their expertise, learning so many new things about the deep sea here with us. We confirmed that this place is a nursery for octopus. We have found a skate nursery. We found a second place of low temperature hydrothermal discharge in this area. More places where we think that seawater is recharging into the seafloor. We're so excited to come back in December and continue this exploration. We're also so excited about all the things we've learned about the deep sea biology and geology of this region to inform conservation and protection in Costa Rica and build awareness and excitement and enthusiasm for all of the amazing creatures we've seen down here. Yeah, so tune in with us back in December. We'll be back making more discoveries, seeing more seafloor, and visiting our octopus friends. Thanks, everybody. Thank you so much to our SOI team for making this happen with us. And thank you so much to all the scientists that have been part of this, both on the ship and on shore, testing out all of our new video annotation approaches. There's so much data here that we're going to be working on over the next few years. So thanks, everybody. Yay!
Bueno, esta fue nuestra última inmersión. Ya vamos hacia la superficie, terminamos esta expedición, pero planeamos volver en diciembre. Y aquí estamos. Esto ha sido 3 semanas extraordinarias conociendo estas regiones de Costa Rica. Esta Costa Rica es conocida. Muchas gracias.